Monitoring cleanliness and hygiene is relatively easy to do on farms using the techniques that are used in the food industry. Protein residue swabs are one tool to evaluate cleanliness. These swabs do not detect bacteria, rather they detect the presence of residual protein left on surfaces. This technique is carried out by removing the swab from its tube or case and swabbing surfaces such as the inside of bottles, pails, rubber nipples, mixing tanks, or any other surface where milk is handled. The swab is then reinserted in its case, the bulb is bent until it snaps, and the reagent is squeezed into the tube. The tube is then slightly swirled to mix the contents. This technique uses a chemical reaction that develops color. Clean surfaces with little or no protein residue result in a green color. Development of a gray color indicates a slight presence of protein residue. If the test develops light or dark purple, it indicates a greater presence of protein residue, which may be the result of improper washing or cleaning procedures. Another technique used to monitor hygiene relies on bioluminescence, or biological reactions that produce light. This technique uses ATP swabs that detect the presence of bacteria. The procedure is the same as outlined for the protein residue swabs, except that this technique requires a special handheld reader called a luminometer. This device measures the brightness of the light produced by the reaction, and it expresses the results in relative light units, or RLU. After swabbing and reinserting the swab in its tube, the reaction develops as soon as the reagent is squeezed out of the bulb. At this point, the whole tube is inserted into the luminometer. The device then takes a reading and displays the resulting RLUs. When bacteria are present, the reaction will produce light. The brighter the reaction, the greater the bacterial presence, and the higher the number on the display. A benchmark for this technique is a reading below 100 RLU, or the lower the better. In summary, clean and dry feeding equipment and instruments help keep healthy calves. Proper washing procedures include water, detergents, and disinfectants. A sanitizer may also be used to reduce bacterial count. Monitoring cleanliness is a relatively easy and simple procedure that can be done with either protein residue swabs or ATP swabs. For protein swabs, the results should develop a green color, and for ATP swabs, the reading should be less than 100, the lower the better. We hope this video provided general guidelines for monitoring hygiene and cleanliness in the calf kitchen.